The purpose of this article is to provide a detailed stepwise visual protocol for extracting aqueous metabolites from cultured cancer cells for subsequent metabolome analysis. The main advantage of this technique is its simplicity and reliability for extracting metabolites from adherent cells. But above all, it's well optimized for metabolome analysis using Capillary Electrophoresis-Mass Spectrometry, or CEMS.
Metabolomics is a powerful technique to identify biomarkers for detecting early-stage cancer and predicting chemotherapy response of cancer patients. We use cancer cells in this demonstration. However, this technique can also be applied to harvest metabolites from other adherent cells such as fibroblasts and iPS cells. Metabolite concentrations are normalized based on the number of viable cells, so careful preparation of culture dishes is the key for obtaining reproducible data. Demonstrating the procedure will be Ami Maruyama, a technician from my laboratory.
To begin, plate HCC827 and PC9 cells in 100 mm dishes containing 10 ml of rPMI 1640 medium supplemented with 10% fetal bovine serum. Place the dishes in an incubator at 5% carbon dioxide and 37 degrees Celsius for 24 hours to culture. After incubation, gently tilt the 100 mm culture dishes to aspirate the cell culture media. Wash cells.